Good evening, Neha. So, good evening, guys. Please share this link with your friends. Okay, so it's already 5 minutes. So, without wasting a time, let's start the lecture. Okay, so in last lecture, we already discussed our plasma membrane and cell wall. Okay, so except of these two organelles, there are few organelles inside of the cell. Not few, many organelles inside of the cell. And today, in our second session, we are going to see what are the organelles inside of our cell. Okay, so good evening and welcome to Unacademy. You are watching the Maharashtra CET examination channel of Unacademy. Before starting the lecture, it is necessary to introduce myself. This is Abhijit Madkar and I have a 6 year of experience with biology in 11th and 12th. I also cracked NET CSIR examination in December 2016 with All India rank 60 from Life Science. Okay, so guys, this was about me. So this is Unacademy, the India's largest platform. If you want to join this platform, please install this app, Unacademy Learning app. Okay, so this app is available uh, on App Store. So if you want to join this, please install this app and sign in with your accounts so you can join this app as early as possible, so you will have the more advantage about your CET and the other examination, because this app is not restricted to CET. There is another courses and the lectures you can see and watch. If there is any favorite educator you will have, so let's join this channel. So this channel on Telegram. So if you don't have this Telegram app, please download this app from your app store and copy this link into the search box of Telegram and then join this channel of Telegram. This channel is for daily updates of lectures. Okay, so here is update of which lecture is held at and what time. Okay, so this channel is for your daily update for Maharashtra CET examination. If you want to use this Unacademy unlimitedly, it is necessary to subscribe us. Okay, so uh, for 12 months, the fee is 16,500. But if you subscribe with this code ABHIJIT, so this capital code will give you a definitely discount in the fees, and the total fee for 12 months is 14,850. And for 24 months, the fee is 24,000, and with discount by adding this code fee is only 21,600. Okay, for 24 months. So your 11 and 12 automatically get discount, and you will have the more lectures on this app. Okay, so guys, this subscription gives you unlimited use. What is mean by unlimited? So here is also a facility for offline lecture. That means you can download the lecture and you can see it offline from this our app. Okay, so there is also a doubt solving session with your favorite educator. So if you want to meet your favorite educator, so or the want to join in a doubt solving session, so you can also join with this a single subscription. Okay, so this single subscription gives you unlimited uh, use of this app. So let's start with our lecture, cell structure and function, part 2. Okay, so in part 1 we already discussed plasma membrane, cell wall, its component, structure and function also. Okay, so we already done with this part. So in this part we are going to discuss about the uh, membrane bound organelles. Okay, so these membrane bound organelles are only with eukaryotic cell, and do not have any membrane bound organelle present in a prokaryotic cell. So prokaryotic cell is always without membrane bound organelle. So okay, so there is no any membrane bound organelle. Okay, so prokaryotic cell do not have any membrane bound organelle. That's why the each and every enzyme exposed to a cytoplasm and cytoplasm exposed to the DNA. So there is no any uh, compartment inside the cell. But in eukaryotic cell, you know that there is a compartment, a different compartment involved in a different function. So that's why there is minimum chances of diffusion of material from one compartment to the another compartment, and uh, it results in less problems in the process of metabolism. Otherwise, what happen in a prokaryotic cell, this type of molecules which is here in more amount, for example, and the metabolism of this molecule is starting here, but because of the high concentration here, this molecule will move toward the low concentration part of a cell, and because of this diffusion it causes a problem with the enzymes or in a cell. So ultimately efficiency of enzyme or efficiency of a cell is completely depending upon the compartments. If there is a compartment, there is a more convenient way to metabolize that particular molecule, or there is also an easy way to metabolize that molecule if it is present in a small part of a sac. Okay, so the first compartment we are going to see is endoplasmic reticulum. Okay, so how endoplasmic reticulum looks like, it is an extension of our outer membrane of nucleus. Okay, so it looks like here, it looks like a tubular structure out of the nucleus, and this tubes called cisternae. Okay, so these are the tubes, we call it a cisternae. Okay, so these tubes automatically get converted into vesicles, and these vesicles moves toward the Golgi complex. Golgi complex is always placed in the front of this endoplasmic reticulum, and this is your endoplasmic reticulum. Okay, we call it simply ER, endoplasmic reticulum. Okay, so this is a network of membranous tubule and sacs, we call it a cisternae. So where is your cisternae? This is a cisternae. So this is a cisternae, which is a tube or sac-like structure of our endoplasmic reticulum. This endoplasmic reticulum forms or contribute more than 50% of total membrane of eukaryotic cell. That means, for example, this is your eukaryotic cell. So this membrane of eukaryotic cell is synthesized, or this membrane is very similar to the membrane of endoplasmic reticulum. Okay, so this is how this endoplasmic reticulum contribute in a cell. But this is not a main function of endoplasmic reticulum. There is another things which are conducted by endoplasmic reticulum, and that's why it is famous for that. For example, a protein synthesis. It is famous for protein synthesis. Okay, why? Let's see. A cytoplasm which is inside of a lumen or inside of a ER cytoplasm. Okay, so what is a laminal cytoplasm? The part of cytoplasm which is inside of the endoplasmic reticulum, we call it a laminal cytoplasm, and the part of endoplasmic reticulum which is not or the cytoplasm which is not part of our endoplasmic reticulum. Okay, so for example, cytoplasm or the liquid which is outside of the endoplasmic reticulum, we called it a non-laminal cytoplasm. Okay, so cytoplasm here out of the endoplasmic reticulum, we called it non-laminal cytoplasm. Let's see, I am drawing here this type of cytoplasm by blue color. So this blue color shows non-laminal cytoplasm out of the endoplasmic reticulum. So you can also distinguish the or you can also classify the cytoplasm in laminal and non-laminal depending upon the ER, endoplasmic reticulum. This tube-like structure is divided into a two parts. Okay, so how they divided and on what basis they divided into the two part? The first thing and the last thing about this structure division is a present of a ribosome. If there is a ribosome, it looks like a rub inside of microscope. Okay, so what guys, is there anything? Okay, is there anything? Please ask. I will definitely answer you. Okay. So the structure which shows a granule-like structure on the membrane of endoplasmic reticulum, we call it a rub endoplasmic reticulum. Why rub? Because it looks like a rub inside of the microscope. And if there is absence of ribosome or that granule-like structure on the ribosome, looks like a granule on membrane. Okay, so if membrane is without granule, it looks like a smooth, very smooth. So that time we called it a smooth endoplasmic reticulum. So that's why I asked one question: why rub and smooth? So why rub? Because it looks like a rub inside of microscope. Okay, so in microscope, the structure are very tiny, very small, very micro in a size. You can't see the structures by your naked eye. Okay, so we can't see a cell also. Cell is too small. We can't see by our naked eyes, and we know that. So these structures are observed inside of microscope, under microscope, and under microscope. It looks like a rub. That means ribosomes are present, and if it looks like a smooth, that means ribosomes are absent. But how can you understand it is endoplasmic reticulum or it is Golgi body, or there is also different type of structure which shows tubule like a appearance? Okay, so let's see how. First of all, let's complete the smooth endoplasmic reticulum. Why it is a smooth? Because it looks like this. Okay, so this is a structure of your smooth endoplasmic reticulum, made up of tubes. Okay, so this is a smooth endoplasmic reticulum. Looks like a smooth inside or under microscope. Okay, these are the vesicles. If I cut it, it will be appear like this. So it's a three-dimensional view. You can say it's a three-dimensional view of endoplasmic reticulum. Okay, so it looks like a smooth. That's why we called it a smooth endoplasmic reticulum. But there is a difference in structure. That means we know that structure is an important part in our biology. If there is a difference in a structure, definitely the function of that particular thing will change. Okay. So let's see first smooth endoplasmic reticulum. Then we will jump to the right rough endoplasmic reticulum. Okay. So it is smooth because of lack of this granule-like structure on the membrane. Okay, lack of granule-like structure on the membrane, and these granule-like structures are the ribosomes. Okay, ribosomes are made up of rRNA. The small r stands for ribosomal RNA. Okay, so R is for ribosome. rRNA plus proteins. So these two biomolecules comes together to form a ribosome. It looks like this. Okay, this is how ribosome looks like. It is made up of two subunit. One is a small subunit and one is a large subunit. Okay, so we will see in detail about ribosome. Now let's see This smooth endoplasmic reticulum is involved in the synthesis of lipid, mostly phospholipids. So phospholipid, what is phospholipid? This is part of our plasma membrane. Okay, so these phospholipids are the part of plasma membrane, and our plasma membrane possesses most of the phospholipid as a part of fluid things. Okay, we already discussed fluid mosaic model. Okay, lipids are responsible for fluidity. So this plasma membrane which shows fluidity, most of the lipids are present in this phospholipid form. Phospholipid looks like this. Okay, this is a glycerol and there is a two chain below. These chains are made up of fatty acids. So those who are absent in last lecture, please see and try to understand. And this is a head group. This head group is always a glycerol. That's why we call it simple lipids are made up of ester. Simple lipids are made up of fatty acid and glycerol. We called it esters of fatty acid and glycerol. Okay, if there is a phosphate group, okay, I am not going to draw a bond here. Phosphate group, it will definitely a phospholipid. Okay, so at that point we call it a phospholipid. If there is again a one bond with a choline molecule, choline contains nitrogen. Okay, so nitrogen containing compound, choline is a nitrogen containing compound. So in this situation we call it a phosphatidyl if this choline is replaced by serine, we call it phosphatidylserine. So depending upon the third group which is on the head of a phosphate, the name of this compound, that is a lipid, is changed with respect to the group which is attached on the head of a phosphate. Okay, and if this phosphate is also absent, we just call it a diacylglycerol. Okay, diacylglycerol, because there is a two fatty acid chain. If there is a one fatty acid chain, we call it a monoacylglycerol, MAG. And if there is a three fatty acid chain, we call it a triacylglycerol, TAG. Okay, so this is how the lipid is present in nature, and this is how we eat it. Okay, so if we eat a TAG, then it is converted into MAG and two fatty acids and then transported by our uh, blood. Okay, so we will see how lipids is digested, and protein and also carbohydrate. So we will have a three different lecture on the digestion of carbohydrate, digestion of lipid and digestion of protein. So you don't worry about that. I will take one more lecture about the digestion of a nucleic acid. Okay, so it is also important as per the your examination. So it is also involved in a metabolism of carbohydrate. Carbohydrate means a sugar. Okay, so this carbohydrate is also called as saccharide, and saccharide means a sugar. So it is involved in the sugar metabolism also. Detoxification of drug and poison is also a job of this endoplasmic reticulum, smooth endoplasmic reticulum. Okay, so simply you can say SER, smooth endoplasmic reticulum. Okay, people also write it like a SER in a short form. Okay, so guys, let's see what is meant by these drugs and poisons. Okay, most of the time when we feel illness, that time we take few drugs. Okay, these drugs attack on microorganism, so that means there is a few groups. Okay, attack on microorganism means there is few chemical groups which are reactive in nature. So it is necessary to stop this reactivity or the inactivate them by our body. Otherwise they will, if they will remain in our body for longer time, definitely they will harm our body also. Okay, so to stop them, to stop this chemical group, it is necessary to deactivate them, and this deactivation you can say it is detoxification. So detoxification doesn't mean this poison to a non-poison. Okay, toxic to a non-toxic. Okay, this is not the way we call it detoxification. Sometimes poisons are not easy to metabolize into the non-poison material. That time this smooth endoplasmic reticulum convert it into a poison to less poison. Okay, and toxic to less toxic. Okay, so let's take an example of this poison and toxic material. So for example, our body when involved in a protein metabolism. Where? In protein metabolism. This protein metabolism result in the one group which is harmful to our body is ammonia. Okay, so ammonia is released from protein metabolism, and it is not easy to hold this ammonia inside of our body. So it is necessary to convert it into a non-toxic material, but not easy to convert into a non-toxic material. That time our body convert it into a urea. Okay, so urea is a less toxic as compared to ammonia, but it is also toxic. Okay, so if there is if we talk about the reptiles, so the reptile convert this urea into a uric acid. Okay, so this ammonia is converted into a uric acid in reptile, and they excrete it. We excrete this urea as a urine. Okay, and if there is again a few organism like a scorpion, the spider in our house, it is hanging on its own resins. Okay, so the spider, scorpion, and then penguin also, these organisms able to convert this a very less toxic material, that is uric acid, into the guanine. Okay, and these organisms are called as a gonotelic organism. Okay, so this is how the conversion of material inside of our body happens. Okay, so if it is impossible or it is very hard for a body particular poison or toxin into the non-toxic material, so that time body just transfer it into the less toxic compound. Okay, so let's see the next function of smooth endoplasmic reticulum is the storage of calcium. Okay, so why calcium? Because guys, this calcium 2 plus, it is ion and it plays important role in signaling. Okay, so it acts as a secondary signal. It's secondary signal molecule. So amplification of signal is required in our cell. For example, cell get any signal from the outside. It is necessary to amplify this signal into a many way, and that time this calcium plays an important role in the signaling process. So it is a need to protect it or store it inside of the uh, membrane bound organelle. You can't leave all the calcium ions inside of the cytoplasm, otherwise it will start something which is not a normal in our body. Okay, so this calcium is again stored inside of the smooth endoplasmic reticulum. We know that in female when they complete its menstruation cycle, or we call it a menopause, a stop of my menstruation cycle. After that, there is a change in hormonal coordination, and it result in this calcium metabolism or malfunctioning in a calcium metabolism, and there is a different type of diseases related to the calcium ion metabolism or the storage of calcium ion. Okay, so calcium ion is important in our body like the other ions also. Okay, but it is necessary to store it inside the sac and not expose to the cytoplasm except it is necessary. Okay, so thank you very much, Ajay sir. So let's see guys, in this smooth and rough endoplasmic reticulum you can see a granule like structure on the rough endoplasmic reticulum. So guys, this is not two different structures. Okay, the same type of tubules come together and they form rough and smooth endoplasmic reticulum. The difference is only present and absent of ribosome on this structure. Okay, so if ribosomes are present, you can say ribosomes are present, then you can say it is a rough endoplasmic reticulum. So I am going to write here in short RER. And if you see this structure do not have any ribosome or granule like structure on it, so how smooth it is, and that's why scientists call it a smooth endoplasmic reticulum. Okay, so why smooth? Absent of ribosome. And why rough? Presence of ribosome. Okay, so this difference is a structural difference, but if you see in a function, this presence of ribosome will change all the function of the smooth endoplasmic reticulum into the rough endoplasmic reticulum, and you can see how rough endoplasmic reticulum act in our cell. So this is our rough endoplasmic reticulum and its function. The presence of ribosome make it a factory of a protein. So what is meant by factory of protein? That means a rough endoplasmic reticulum is involved in a protein synthesis. It is involved in protein synthesis, guys. Okay, so friends, see how only a presence of ribosome will change the complete thing about endoplasmic reticulum. How? Because this ribosome, what I am drawing is a ribosome. Okay, it looks like a small and large subunit. Okay, we already discussed. So there is a gap between two subunit which is for holding mRNA. Okay, so this is mRNA, messenger RNA. It is the result of your transcription from a DNA. So what is the information we got from our parent is converted into mRNA, and then this information decoded. mRNA is decoded into a protein so that protein can work properly. Okay, so proteins are worker in our body. DNA knows everything, but DNA does not do any work in our body. That's why it is necessary to convert this information of DNA into the worker molecule, that is protein, and it is mediated by RNA, and that type of RNA we call it mRNA. Okay, so this gap is for mRNA, and this mRNA is decoded into the protein by this guy. We call it a ribosome. Okay. So this guy is responsible for the translation process. Okay, so this is a transcription. Transcription means conversion of DNA into the RNA, and conversion of RNA into the protein is called as a translation. And translation is done in the presence of ribosome, so that the presence of ribosome made this tubular structure of endoplasmic reticulum as a protein factory. Okay, so why this is rough endoplasmic reticulum is called as protein factory? Because of only presence of ribosomes. Only presence of ribosome is responsible for the rough endoplasmic reticulum. We call it a protein factory. Okay, so this is a main reason we call it a protein factory, presence of ribosome, because it is involved in a synthesis of protein. Okay, so let's see. Rough endoplasmic reticulum is always attached to the nucleus by its one side. Okay, one side means, for example, this is nucleus, and I'm drawing here nucleus, outer structure, outer membrane of nucleus. Okay, so the rough endoplasmic reticulum attached it like this. And in endoplasmic reticulum, these all cisternae are connected to each other. Okay, so there is a gap between two cisternae, or the canal between two cisternae, from which the material can transport. So it looks like a presence of dotted structure on this. So I'm going to make it 3D. So if it is 3D, it looks like this. Okay, guys, wall or the membrane of rough endoplasmic reticulum. If let's take an example of this ribosome which is present on the structure. So for example, this is attached to this wall. Okay, so this is attached to this wall. From the behind of wall, it will result in the formation of protein inside of the membrane. So protein is now inside of the membrane. Okay, so this is how protein is synthesized inside of the rough endoplasmic reticulum with the help of ribosome. That's why we call it a factory of a protein. Okay, so transport vesicles involve the transport of protein. That means this protein is then transported by this rough endoplasmic reticulum like this. For example, this is our rough endoplasmic reticulum, and it is going to form a vesicle here. So this is vesicle which is going to detach from rough endoplasmic reticulum, and it possesses inside a protein. Okay, so proteins are synthesized into the rough endoplasmic reticulum and they are packed into the sac like a structure. We call it a vesicle. So this is our vesicle, guys. From now, this is not a sac. We call it a vesicle. Okay, these vesicles leave endoplasmic reticulum and moves toward the Golgi complex. Golgi complex for what? For the further processes and sorting. Okay, so sorting and further process or modifications is a job of Golgi complex. So the first job of rough endoplasmic reticulum is the synthesizing protein, and one more thing we can add into the rough endoplasmic reticulum, a protein synthesis plus the folding of protein. Okay, so folding of protein is also a part of your rough endoplasmic reticulum. There is a chaperone proteins which are involved in a folding of protein. Okay, chaperone start from ch. Okay, chaperone. Okay, so che, the spelling is started from che. Sometimes people pronounce it like a chaperone protein. Okay, so both pronunciations are true in this sense because the both proteins are involved in a folding of proteins. Okay, so what is meant by folding of protein? We know that protein is a chain of amino acid, but you can't leave this chain in a cytoplasm as it is because there is a particular shape of a protein. It may be like this or maybe different. Okay, but to understand us, we made it like a cartoon. This. Okay, so I always draw your enzyme like this. Okay, so you can see there is a mouth or the active side of enzyme which can involve or which can take part spontaneously inside of the chemical reaction. Okay, so this is how we draw it, but actually the structure of a protein is made like threads which is gathered together. Okay, so this is how our protein looks like, and this is how we draw it into the cartoon form. Okay, to increase our understanding. So these are the functions related to the rough endoplasmic reticulum. Let's see, guys. This is a rough endoplasmic reticulum, smooth endoplasmic reticulum, Golgi complex, and your plasma membrane inside a single diagram. And now just discuss this diagram in detail. Okay, so this is a nucleus. So this nucleus is extended, or the membrane of a nucleus is extended into the endoplasmic reticulum. This is endoplasmic reticulum. This is endoplasmic reticulum. Okay, so I'm going to write here ER, endoplasmic reticulum. But presence of this granule like structure, that is ribosome on ER, made it RER, and smooth without any roughness or the presence of granule like structure on this made smooth endoplasmic reticulum. Okay, so absence of ribosome made these tubules into the smooth endoplasmic reticulum, and the presence of ribosome made it a rough endoplasmic reticulum. So these are the two structure of endoplasmic reticulum. So when a protein is synthesized, so these are the proteins which are synthesized inside of the membrane or the rough endoplasmic reticulum. These proteins are now here, you can see they are going to packed inside of the vesicle. This vesicle is a move forward and moving toward the Golgi complex. When this vesicle comes to a Golgi complex, it's attached to the cisternae or the tubules of the Golgi complex. Okay, so by which phase or which side the vesicle which is came from the rough endoplasmic reticulum and attached to the Golgi complex, this side or this phase is called as cis phase or cis region of a Golgi complex. Okay, for example, this is your endoplasmic reticulum. Okay, so I am drawing here roughly, and this is your Golgi complex. So this is a phase of Golgi complex which is toward the rough endoplasmic reticulum, so we call it a cis phase, and the phase which is opposite to the RER or the ER, endoplasmic reticulum, we call it the trans phase. transphase or trans region. Okay, so these are the two regions which contribute in the structure of a Golgi complex, so that we gain information of Golgi complex which phase is toward the rough endoplasmic reticulum and which phase is opposite to rough endoplasmic reticulum. So the phase which is opposite to the rough endoplasmic reticulum, we always call it a transphase, and this transphase always toward the plasma membrane. Okay, so the proteins which are synthesized by rough endoplasmic reticulum are always move toward the Golgi complex. For what? For the sorting and the modifications. Okay, so this is how Golgi complex absorb the proteins or accept the proteins which are synthesized by our rough endoplasmic reticulum, and then these proteins again packed inside of the vesicle and move toward the target place. Okay, so whatever target is, for example, this protein is targeted or made for the mitochondria, so it will target it toward the mitochondria. For example, this protein is a protein of chloroplast in a plant cell, it will move toward the chloroplast in a plant cell. For example, this protein is a part of a lysosome, it will move toward the lysosome in an animal cell. So this is how the proteins are sorted or move toward the target place, and it is because of the Golgi complex. Okay, so you can't say endoplasmic reticulum sent protein toward the target, because endoplasmic reticulum do not have any facility to read the address on that particular protein and then target it toward the its destiny. Okay, so the destiny of a protein is decided by the sequence which is present on the protein, and that sequence also act as an address for the protein, and then this address or reading of this address is a facility of our Golgi complex, so that Golgi complex is involved in sorting of protein, and that's why we called it a transport system of a cell. Okay, post office म्हणतो की नाही आपण त्याला, it is a transport system of our cell. We generally called it a post office of a cell. In school days we called it a post office or a transport system because of the facility which involved in sorting of a protein. Okay, so each and every time when the protein is sorted by this Golgi complex, it always tag it by the protein or the other biomolecules, and these molecules or tag is called as marker. Okay, so marker is there, marker decide where the protein will move, and these protein are transported by the motor proteins. They just pick this vesicle and move toward the target. Okay, so these motor proteins are also important. We will discuss this motor protein in a different lecture. Okay, so let's see what is Golgi complex and how it act in our cell. So Golgi complex is also called as Golgi apparatus or Golgi body. Okay, so this Golgi word came from the name of scientist. Okay, so this is a name of scientist. So this is a Camillo Golgi. Camillo Golgi is a scientist who explained Golgi complex. Okay, Camillo Golgi is a not a first man who discovered the Golgi complex. Okay, so discovery of Golgi complex is by another scientist before the Golgi, and then Golgi comes into focus when Camillo Golgi explain it beautifully. Okay, so when you see the name of scientist for any particular structure or any particular equation or any particular thing in a science, that time it is not each and every time correct that that particular structure or that thing is discovered by scientist. Okay, so this is not correct, my dear friend. Most of the time the structure is explained, or the function or the equation or the whatever it is in a science is explained by that scientist in such a beautiful way that that particular structure is recognized by the name of that scientist. Okay, so there are many examples like this. For example, Watson and Crick. Okay, Watson and Crick never discovered the DNA. They are responsible for the best explanation of double-stranded helical structure of a DNA. Okay, so DNA is discovered by another scientist who was a doctor, and he saw the precipitation on the spirit swab. Okay, so a spirit, not swab, but what he does, he just swab the wound of a people and drop that cotton inside of the spirit, and he just see the precipitation in the spirit, and that's why he check this chemical, what is this chemical, and he called it uh, nucleic acid. Okay, so this is how the DNA is discovered by a doctor, and it is explained by Watson and Crick. So that's why we call it the Watson and Crick. But the discovery is not with Watson and Crick. So the same thing is happening here. This discovery of Golgi complex is not with the Camillo Golgi. Okay, so guys, this is the thing with Camillo Golgi. Please write these 2 points. These are the necessary: manufacturing cum packaging and transport unit of a cell. Okay, so Golgi complex is manufacturing cum and transport unit of a cell. So please write this point. Is there anything? Please ask me. Is there anything you are not getting? Please ask me, my dear friends. Okay. So let's see. Golgi complex consists of stack of membranous sac. We call it a cisternae. Okay. The structure of Golgi complex is looks like a structure of endoplasmic reticulum. So there is no big difference between two structure except of the linking of these cisternaes to each other. Okay. So it will looks like this. Okay. So if you draw it, so this is how your Golgi complex looks like. So these are the structures which are, or these are the vesicles which are leaving a Golgi complex from the transphase. Okay, and here is a structure which is accepted by the Golgi complex coming from the rough endoplasmic reticulum. So these structures are also vesicles which is get attached to the cisternae of a Golgi complex. Okay, so this is how our Golgi complex looks like. If you see the structure of endoplasmic reticulum, it is also same, but there is a small difference. Endoplasmic reticulum is always attached to the next cisternae, and there is a canal between the two cisternaes in a in endoplasmic reticulum, but this canal is absent in a Golgi complex. Okay, so this is how these two structures are distinguished. So here is always a canal. Okay, so I am going to draw it by red color. Guys, please see, this is a canal which connect all cisternae, and this is the difference between a two structure. Otherwise, if you see, both structure are made up of tube like structures. Okay, and we call it a cisternae. The cisternae in a Golgi complex are not physically connected to each other like ER. Okay, so that's why I write here there is a difference between endoplasmic reticulum and Golgi complex is a major difference, like the connected tubes are present in endoplasmic reticulum, and Golgi complex do not have any connection between the different cisternae or tubes. The diameter of cisternae varies from 0.5 micrometer to the 1 micrometer. So this micrometer, what is meant by micrometer? It is 1,000th part of your millimeter. Okay, so 1 millimeter is equal to 1,000 micrometer, you can say. Size of Golgi complex inside of a cell. Okay, so this size means inside of a cell. How small and how large Golgi complex will be present inside of a cell is completely depending upon the function of a cell. It is depending upon the function of cell. So what is meant by function of a cell? For example, let's see role of a Golgi complex. It is involved in the sorting of a protein. Okay, that means, for example, here is a cell. I am taking this cell as an example. So in this cell, it is involved in a secretion. Okay, so for example, gland cell. Glands are involved in a secretion. So for the secretion, we required more vesicles for a fast and a large secretion of product. We required more vesicles. That time we required a large Golgi complex inside of a cell. Okay, so if it is uh, if this secretion is a protein made or based on the protein, there is also a large uh, endoplasmic reticulum present. Okay, if this secretion is a lipid based which is synthesized by smooth endoplasmic reticulum, that time we will have a large structure of smooth endoplasmic plasmic reticulum inside of a cell. Okay, so the structures or the number or the size of these organelles is completely depending upon the function of a cell. Okay, so they, if they are involved, for example, let's take example of smooth endoplasmic reticulum. Smooth endoplasmic reticulum is involved in a detoxification of material or the conversion of toxic or poisonous material into the less toxic or less poisonous material. So which organ or which cells are mostly involved in this function are the liver cells. Okay, so our liver cells shows a dominancy of smooth endoplasmic reticulum. You can say smooth endoplasmic reticulum is a large in a size as compared to the other cell. It is a larger in a liver cell. Liver cell generally we call this hepatic cell. Okay, so this is how your looks like. This is how your liver looks like, and this is involved in the detoxification. So there is a more number or the large structure of smooth endoplasmic reticulum as compared to the other cells. Okay, so this is how the structure of these organelles is decided by the function of a cell. That's why I always said that the structure is a reason behind the function. Okay, if you got a structure of particular cell, you can calculate this cell is involved probably in the function of dash dash. Okay, depending upon the information you gain from the study of that particular cell. Okay, is there anything? Yes. The face toward the endoplasmic reticulum, we call it a cis face, and the face which is opposite to the endoplasmic reticulum, we call it a trans face, and this is giving us idea about the location. So that's why, guys, the Golgi complex is always in front of ER. Endoplasmic reticulum is here. That means these vesicles are coming toward the Golgi complex. So Golgi complex is always placed in front of our endoplasmic reticulum. Okay, so this is how the arrangement of Golgi complex and endoplasmic reticulum is managed by our cell. Okay, so the function of Golgi complex. The first function of Golgi complex is a modification in secretion of endoplasmic reticulum. So any molecule or protein which is coming from the endoplasmic reticulum, it may be modified by a Golgi complex if required. Okay, so quality of that product is also checked by the Golgi complex. For example, this product is came from the rough endoplasmic reticulum or endoplasmic reticulum. So RER, we are taking an example of protein, and this protein had address in the form of a sequence of amino acid. So when Golgi complex accept this protein, the first this protein is go or move through the process of checking, and in that process, if Golgi complex cut, this protein is not properly folded. Okay, so it should be a properly folded. We know if there is a change in a structure. That means if improper folding definitely shows a different shape or different structure of a protein. That's why if there is any change, Golgi complex recognize it will again send this protein toward the rough endoplasmic reticulum for proper folding, and then endoplasmic reticulum make few changes and again send toward the Golgi complex. So it happens 2 or 3 times, guys. After that, this protein again come to the Golgi complex with a misfolded conformation. That time Golgi complex again send it to the RER, and then RER cut this protein into the amino acid. That means degrade this protein. So protein has only 2 chances of modification. After that, they will degrade and synthesized in a new way by rough endoplasmic reticulum. So this checking and maintaining the quality of a product is also function of a Golgi complex. Okay, so Golgi complex also play a main role in sorting of a protein. So what is meant by sorting? For example, this protein which came from the rough endoplasmic reticulum, Golgi complex study it, and then after studying Golgi complex, what it like, this is a protein of lysosome. Okay, so Golgi complex send it toward the lysosome. For example, this protein is of mitochondria, then Golgi complex send it to the mitochondria. If it is a protein of plasma membrane, Golgi complex send it to the plasma membrane and placed in a plasma membrane. For example, this is a plasma membrane. So send it and manage or arrange it into the proper manner here. So it can take information from outer part of a cell. Okay, for example, this protein is a secretory protein which do not going to live inside of a cell and wants to move, or it is necessary to secrete it into the blood. That time Golgi complex secrete it into the out of the cell. Okay, so this type of a job is done by Golgi complex. That's why we call it a transport system. Golgi complex always kept a marker on the vesicle to identify their targets. Okay, so this vesicle always comes with a marker toward the target place. Okay, so there is a marker on the target place. This marker is recognized by lysosome, and then lysosome take this protein inside of the organ. Okay, so lysosome is a organelle. We know this is uni or the single cell organelle. Sorry, a single walled organelle, single membrane organelle. It is not surrounded by 2 membrane. It is surrounded by 1 membrane. So it will take this protein inside. Okay, so same, there is a different marker for the mitochondria. So mitochondria recognize this marker and take it as a part of a mitochondria. Otherwise, these vesicles are sent again toward the Golgi complex if there is any changes or any miss marking. Okay. Golgi complex synthesize its one product like a pectin in a plant. So pectin in a plant is synthesized by Golgi complex. So Golgi complex also involved in a few type of product synthesis. It is also involved in the production of glycoprotein. So what is meant by glycoprotein? Glyco means carbohydrate and protein means protein, amino acid or chain of amino acid. So this type of proteins are synthesized by Golgi complex. It doesn't mean this protein part is also synthesized by Golgi complex. This protein part is synthesized by endoplasmic reticulum and then sent toward the Golgi complex. Then Golgi complex add this saccharide unit or sugar unit on the protein, and this modified protein is again sent toward the target place. For example, our blood group contains uh, carbohydrate as a structure which is exposed toward the blood, okay, or out of the cell. This is also a saccharide or carbohydrate structure. So our blood group is also made up of carbohydrate structure. Okay, so let's see, guys. This is again structure of endoplasmic reticulum, Golgi complex, and the secretory system inside in a single diagram. So this is your nucleus. You can see the extension of the membrane of your nucleus is called as RER, rough endoplasmic reticulum. Okay, so this is rough endoplasmic reticulum, which is going to make or synthesize protein and then send it toward the Golgi complex, and then this protein moves toward the target place. So target is always leave Golgi complex from transphase, and vesicle always come inside of the Golgi complex from a cis phase. Okay, so this vesicle is came from the endoplasmic reticulum as a transport vesicle. Okay, sir, after degradation, where does the protein go? So let's see, guys. For example, this protein is misfolded. Okay, so Golgi complex again sent it toward the rough endoplasmic reticulum. Then rough endoplasmic reticulum again refold it and send it to the Golgi complex. Still, there is any problem inside of the structure of a protein. That time, this rough endoplasmic reticulum sent this protein toward the uh, protease complex. Okay, so protease complex is let's see, this is cartoon. I'm going to draw a cartoon of protease. So this protease is a mixer like structure. For example, there is a fan inside. Okay, it will take a protein from one side and the amino acid from other side. So this amino acid is not excretory or the or worst part, or the we can say a waste part, which is excreted by the cell. This amino acid is a raw material for protein. So this amino acid again used to synthesize the protein. Okay, got it, guys? Who asked this question? Got it? So these pieces or the amino acid, pieces of protein or the amino acid can use by the cell for the new synthesis of a protein. Okay, so there is no any changes in amino acid after degradation. Amino acids are amino acids after degradation and before degradation also. Okay, so this type of system is running inside of our body. Okay, let's see what is the role of lysosome. Lysosome, we call it a suicide bag. Okay, so it is an interesting part which is inside of our cell. Let's see, for example, this is a cell, okay, which is surrounded by a different cells. So one cell, we know that, which is surrounded by a different cell in our body. So this group of cell, we call it a tissue. For example, I am talking about tissue, or I am taking just a few cells from the tissue. For example, let's take an example of corona. Okay, so the most famous virus in the world now. Okay, so this is a corona which is going to infect this middle cell. Okay, so middle cell start fighting with the corona, but after a few days or after a time spending in fighting, this cell understand that I am not going to win this war, and that time this cell signal to the another cell that I am infected so badly, before within a few hours or within a few days I will burst. Okay, so this type of signal, when this cell secretes inside of the environment or in the vicinity of a cell, that time our immune system comes toward the cell. Human system comes toward the cell, and the special cell, which is we call it cytotoxic T cell, will come and signal the cell on this receptor, please suicide. Okay, so that time, in the result of this signal, will get activation of lysosome, and this lysosome will get activated. This lysosome starts the pathway, we call it a programmed cell death or the apoptosis. Apoptosis and programmed cell death, both are the same. So this programmed cell death pathway is started, and in this pathway, cell is going to suicide in this way. So this is our cell, and it is now converted into the small sacs like this, and this is a cell. Again there is a new vesicle coming from the cell, and there is again a vesicles, small vesicles coming from the cell. So this cell, which is an intact cell, now you can see it into the pieces of vesicles, and these vesicles are again engulfed by the macrophages. We call it macrophage cell, which is a cell of immune system which can engulf this type of material and again fuse this material with a lysosome and then lysosome again digest it. Is there anything remain from the digestion? Okay, so this uh, system we call it apoptosis. It is a protective system to protect these cells. Okay, so for what happen, this cell do not send anything to immune system or not send any signal to the immune system. That time this cell is burst by our viruses. Okay, so virus going to burst it after the completion of growth of virus. At that time what happened, this virus come outside of the cell and again infect the cells which are present in the vicinity of the this infected cell, and because of that infection will increase with respect to time. But to avoid this infection, our system had this apoptosis pathway by which cell can kill or digest itself completely so that the infection will not spread inside of our body. Okay, killer T cell, Aditya, very good. Cytotoxic T cell and killer T cells are there. Okay, very good guys. There is also one cell, helper T cell. Okay, we will discuss into the uh, immune part of our human diseases a chapter. Okay, so this cell is also interesting cell which is infected by HIV. Okay, so this is a target cell in a AIDS. HIV virus infect this cell. This is considered as a heart of immune system. Heart of immune system. Okay, so this is most interesting topic. I love this topic. Whenever I um, teach, we will see it in a detail. Okay, so lysosome, where it located, it is a part of a cytoplasm. So most of the things about lysosome, I think you are getting. Okay, so please, is there anything? Please ask. Uh, A lysosome contains hydrolytic enzyme which can digest anything inside of a cell at a low pH or the acidic pH. So lysosomal enzyme will only functions when there is acidic pH. Okay, so they are involved in the digestion of macromolecules. So if there is a bacteria, it is also completely digested by our lysosome. If there is a virus, it is also digested by a lysosome. Okay, so if lysosome got anything, it will digest completely. Okay, it also digests a cellular waste. So is there anything a cellular waste inside the cell, it is also sent toward the lysosome so that it can be digested by the lysosome. Okay, so let's see what is there inside of a cell, what is the waste material. So let's say these words are important for the MCQs type questions: autophagy and autolysis. Okay, so when you say it is autophagy and when you say it is autolysis, when for example let's take a cell. In this cell, this is any cell organelle, whatever it is, mitochondria, chloroplast, or whatever it is. Also, okay, sorry, do not consider chloroplast because lysosome is absent in a plant cell. Okay, for example, it is mitochondria, or it is a Golgi complex, or it is another thing inside of the cell and not going to function properly. That is a malfunctioning. So malfunctioning of any organelle is not entertained by our cell, and at that time this malfunctioning organelle is fused with a lysosome like this. Now this malfunctioning organelle is fused with a lysosome. Lysosome will completely digest this organelle and ask cell to make them a new one. Okay, so when our cell organelles are old, get old, or there is anything which is missing in a cell organelle, or there is a damage in a cell organelle. So because of these different reasons, cell organelle will not going to function properly, and these malfunctional cell organelles are digested by a lysosome. Okay, so this is also a role of lysosome. And when we discuss the example of corona in this discussion or in this example, the complete cell is digested with the help of a lysosome, and there is a one more thing which is important, that is a mitochondria. Mitochondria is also equally contribute in the apoptosis or programmed cell death. Okay, so in this situation, when a cell is completely going to digest itself, that time you can say autolysis. And when a complete organelle any organelle of a cell is going to digest by a lysosome, that time you can say autophagy. Okay, so autophagy and autolysis, these are the two words differently used with a lysosome. Okay, are you getting my point here? Lysosome digests bacteria, virus, cellular waste. Then how lysosome does not get damaged? Okay, a very good question. So guys, let's see. This green color name is a power of lysosome hydrolytic enzymes. Okay, so these hydrolytic enzymes, lysosome use these enzymes and digest anything. When these enzymes get damaged or lysosome have need lysosome, for example, lysosome need more enzymes, that time lysosome is filled by the enzymes with the help of rough endoplasmic reticulum. So rough endoplasmic reticulum send enzymes toward the lysosome. Lysosome use it. So there is nothing to digest. Sorry, there is nothing to damage inside of a lysosome. Okay, so lysosome is made for that, guys. Lysosome is made for that, to digest anything. Okay, these organelles, different type of organelles, are not made for the digestion of anything. Okay, lysozyme in eyes is produced by lysosome. Okay, so lysozyme in eyes, it is enzyme, or you can say it is a group of enzyme. Okay, so it is a group of enzyme which is, we generally called it antimicrobial. Okay, so Rajesh, it is also present, this antimicrobial set of enzyme or lysozyme is also present in your saliva. Okay, so they are differently synthesized in the secretory cell. Okay, because these products are mostly secretions. Okay, these are the product of secretions. They are synthesized differently inside of a cell and then secreted as antimicrobial enzymes, or you can say lysozymes. Okay, so these lysozymes are different. Okay, the discovery of lysozyme is also a serendipitous, means accidental discovery. Okay, so scientists which is working with gel page, I think, no, it's a electrophoresis gel, and that time a drop of his tear is poured on the gel, and there is a marking of that drop on gel. That's why he just discovered, or the pursued that thing, and he found that there is a lysozymes. Okay, so this is a story. If you have a time, please read this type of stories. Okay, they are present anywhere. Most of the time, if you join any group on Facebook related to the science, there is also, you can have this type of stories. Okay, these are the interesting these stories uh, help you to memorize the things. Okay, so read more and more stories. What is the function of lysosome? This is how the lysosome function. For example, this is foreign cell or material which is going to digest by cell. That time this is engulfed by cell by the phagocytosis. This type of engulfing or eating is called as phagocytosis, and then this phagocytosis vesicle is joined with the vesicle, that is a lysosome. So this joining is seems like this. Okay, so this vesicle is now here and joined with a lysosome. So this vesicle is called as phagosome because it is taken inside by the process of phagocytosis, and now this lysosome is joined with the phagosome. That time you can say this vesicle is a phagolysosome. Phagolysosome. Okay, so after a few or after a time, this phagolysosome is looks like this. Okay, so what happened? This phagolysosome is completely digested by lysosome. Okay, so the material which is phagocyted by cell is completely digested, and now this material is uh, this material also excrete by the cell into the excretory system, or if there is anything which is important for our body, it is also used by our system. Okay, so this is how our system works, and this is how our lysosome function in our body. So it may be anything, a foreign particle which is trying to invade our immune system, or the damaged organelle, or the complete cell which is infected and not going to move further in the fighting with that infection. So that time lysosome plays an important role. Okay, so the discovery of lysosome is by two scientists. One is Didu, and this scientist, sorry, one scientist, Didu, and this scientist got a Nobel Prize in medicine in 1974 for the discovery of a lysosome. Okay, so enzyme of a lysosome are synthesized by endoplasmic reticulum and transported toward the Golgi, and then Golgi send it toward the lysosome, because endoplasmic reticulum do not have facility to read the address of enzyme and direct a transport. Okay, so any enzyme in or any protein inside of our body will not directly transported toward the target place by endoplasmic reticulum. There is a need of Golgi complex. That's why we call it a transport system of our cell. Okay, it is just fusion. Okay, Prajakta, it's just fusion of two membranes, sorry, two vesicles. So lysosome is also considered as a main player of apoptosis or programmed cell death. This apoptosis or programmed cell death also involved a mitochondria. Okay, why mitochondria is involved? We know that mitochondria is not part of our system. It is symbiotically living with our system. Symbiotic relation. We call it endosymbiotic theory. Okay, so there is also a need a permission of mitochondria to digest the cell. Okay, because in our cell, mitochondria has a few rights because of their DNA present inside of mitochondria. It also त्याला सुद्धा काय अधिकार आहे. Okay, so our cell cannot decide to die without asking a mitochondria. So mitochondrial permission is also important. So lysosome work with the mitochondria, and then cell will enter into the apoptosis. Okay, so lysosome is another organelle except of mitochondria. Except of mitochondria, lysosome can use oxygen. Okay, so the facility of using oxygen is only with the mitochondria except of lysosome. That means lysosome is a second organelle in our body, in our cell, which can use oxygen. So this oxygen is used by a lysosome to synthesize a ROS, reactive oxygen species. Okay, so these reactive oxygen species are made to digest or to lyse the material or the bacteria or the virus or the cell which is trying to invade our immune system. Okay, so immune system मध्ये, for example, let's take an example, a bacteria is trying to invade our immune system. That time bacteria is engulfed by macrophages, and few amount of oxygen is also used by this lysosome to synthesize ROS, reactive oxygen species. It may be then superoxide anion. Okay, so this is our superoxide anion. We denoted by a star. Okay, excited oxygen, or there may be a S2O2 peroxide. Okay, so the hydrogen peroxide is also always there, is a product of lysosome. after using oxygen. Okay. So when oxygen is used by a lysosome, we call it a respiratory burst. A respiratory burst. So respiratory burst means a process by which a lysosome use oxygen. So in our body, the use of using oxygen or the facility for the oxygen use is only with the mitochondria, and then second number is lysosome. It use our oxygen but not continuously. It only use at the time when this lysosome want to digest things inside the phagosome. Why do lysosome use oxygen? Because to synthesize the ROS. Okay, so when lysosome do not have enough machinery, or sometime lysosome has enough machinery, still it is necessary to digest or to lyse that particular bacteria within a minute or very few time. That time ROS helps to digest that bacteria. Okay, so these reactive oxygen species are highly reactive so that they can digest or make a hole in the membrane, and they lost the integrity of that bacteria, and because of that it is easy to digest that bacteria. Okay, so that's why lysosome uses oxygen to synthesize the ROS, reactive oxygen species. Okay, so what is apoptosis? We already discussed, and the role of apoptosis in embryo. Okay, development of embryo. For example, in embryo, the hand is synthesized like this in first phase, and then the cells which are between two fingers are going to die by the process of lysosome. Okay, and then we will have five different fingers in our hand. Okay, so a shaping of a body in the development of embryo is an important thing, and this important thing is also done by this lysosome. For example, let's take another example. At the time when a particular organism wants to change its phase of life, that time also a lysosome plays important role. For example, in a frog had a tail in its early phase of development, but if you see a mature frog, it is without tail. So this tail is removed by the action of a lysosome. Okay, so this is how lysosome involved in the development process also. Okay, so Tay-Sachs disease, it is important to discuss here. Guys, lysosome also secretes a lipase enzyme inside the cell or inside the our body. So this lipase enzyme, if there is any problem with a lysosome which results in a low concentration of lipase enzyme inside of a body, that time the lipids, okay, lipids are the substrate for lipase enzyme. Lipase enzyme just take a lipid and convert them into the uh, glycerol and fatty acids. Okay, so that time if lipase enzymes are low in amount in our body because of low secretion of lipase by a lysosome, that time these lipids are gathered and accumulate into the brain. Okay, and there is impairing in a brain. It results in a what we call it, impairing in a brain. That means it results in a malfunctioning of neurons, or there is a damage in neurons of a brain. Okay, so because of that, this is necessary to release adequate amount of lipase inside of our body, and if this lipase is not secreted by a lysosome with a good or enough concentration, that time our brain is get damaged, and this disease we call it a Tay-Sachs disease. Tay-Sachs disease. It is because of the lipase enzyme which is low secreted by our lysosome. Okay, yeah, maybe there is a baby without finger. Okay, sometime baby comes with a attached joint fingers like this. and after few days a doctor operate it and make these all fingers separate. Okay, so that time lysosome do not play important or the main role between two fingers, or not play its role properly, so it results in a patching of fingers like this. Okay, why it is named a T-Sagdis? Okay, so it may be the name of scientist. Okay, so it's a name of scientist. He discovered it first, so that's why we called it like this. Okay, good questions. Your questions are like the graduate students, guys. So it seems that you do study very well. Okay, so please increase your speed of study. It will again give you a better result. Okay, because more the revision, more you will have a mark in examination. Okay, so acrosome, let's see what is acrosome. Most of the people know, or they can able to remind the structure of acrosome and where it is placed. Okay, so let's see, this is a structure of a sperm, for example. This is how our sperm looks like. Okay, so this part is called as head, this middle part is called as midpiece. Okay, and this is a tail of a sperm. Okay, so in this head there is a one sac at the front of the head. Okay, then here is a centriole, then here is your a nucleus, and again second centriole is here. So one is a distal centriole and a proximal centriole, we know that. Okay, or if you don't know, we will discuss in a reproduction chapter. Okay, so this is the acrosome. It is necessary to digest the all of ova. Ova means egg. Okay, so female ova, it is necessary to digest the all of ova, otherwise sperm will not going to enter, or this 23 chromosome will not going to enter inside of the ova. So the fusion process, we call it fertilization or fusion between two membrane, it requires acrosome as a mediator, and this acrosomal enzymes or this acrosome is considered as um, modification of lysosome and endoplasmic reticulum. Hyaluronidase enzyme, very good. मित्रांनो सगळं लक्षात आहे तुमच्या. Pertilene and antipertilene, it is involved in a fusion of a membrane. Very good, Rajesh. Okay, so one of the pertilene and antipertilene is present on the sperm, and the second one is present on the ova. Okay, so these are the marker proteins present on the ova and sperm, responsible for the species to species fertilization. Okay, म्हणजे ज्या वेळेस पण एखादा ओवा एखाद्या स्पर्मला फर्टिलायझेशनची परमिशन देतो, बिफोर द परमिशन it check that the sperm is from same species or not. Okay, so this is how pertilene and antipertilene works inside our the process of fertilization. Okay, hyaluronidase is an enzyme which is the main part of our acrosome. Okay, so very good Aditya, Rajesh, Prajakta. अरे हुशार आहात तुम्ही, भरपूर अभ्यास करून ठेवलेला आहे. असे पटकन लक्षात आलं पाहिजेत सगळ्या गोष्टी. So now let's try to take this disease, Tay-Sachs disease, inside in your mind somewhere. Okay, so this is also important. Maybe there is a question on this Tay-Sachs disease in examination, upcoming examination. What if it is not from the same species? That time fertilization will not going to proceed, my dear. Okay, so fertilization will not going to proceed. So this is a power of fertilization. Okay, it always check the sperm or the ova are from the same species. That's why it is a species specific. Otherwise there may be different type of organism mate with each other, and the different type of progenies we will have. Okay, but in nature it is not allowed by nature to fuse two different species together. That's why the liger Okay, so liger is a combination of lion and tiger. It is infertile in a nature because we made it inside of our lab. Okay, if you try to fuse the sperm from the tiger and the ova from a lion, it is not going to fuse. Okay, so it required your techniques which is used in a lab. We will study this technique in a biotechnology in this session. Okay, so I arrange this session mostly at the Saturday and Sunday. Okay, so we can take a lecture of more than 2 hours. It requires more than 2 hours for a single subtopic. That's why I arrange it Saturday and Sunday. So guys, do not miss these lectures on Saturday and Sunday. We will discuss biotechnology after completion of our genetic, that is transcription, translation and replication. So we already complete with the replication. Okay, so let's see vacuoles. These vacuoles are present in plant cell mostly. Okay, and they are rare in animal, so it is a part of it, they are famous in a plant. So they may be a larger or smaller depending upon the requirement of the plant cell. If they are larger in a plant cell, that time we call it a central vacuole. Okay, so sometimes they occupy a 90% of cytoplasm. That means central vacuole can occupy the 90% of cytoplasm. In that condition, what happens? For example, this is a plant cell and this is a vacuole inside of the plant cell. It occupies more space, and the complete cytoplasm and the other nucleus and the remaining things are pushed in the corner of a cell, and the remaining portion is covered by the vacuoles. This vacuole, what it store inside? Okay, let's say the boundary of a vacuole is covered by the membrane, and we call it a tonoplast. Okay, so vacuole, the membrane of a vacuole is called as a tonoplast. Okay, so you can see here inside of the a beautiful diagram, this is a tonoplast, a membrane of vacuole, we call it a tonoplast. The sap which is inside of the vacuole is hypertonic to cytoplasm. Okay, so what is meant by hypertonic and hypotonic and isotonic? So these are the concept related to the tonicity. Hypertonic means a more concentration of solute. Okay, so this shows a more concentration of solute as compared to the cytoplasm. That's why we call it hypertonic to cytoplasm. If I said it is a hypotonic to cytoplasm, that means a low concentration of solute inside of the material as compared to the cytoplasm. Or you can say there is a more thing which is always inside of the vacuole is a water. Okay, so here is a water also placed inside of the vacuole. Okay, so this water applies a pressure on the vacuole and we call it a turgor pressure. Turgor pressure. So turgor pressure means a pressure of water which is applied on the wall. Okay, so in a plant there is a problem with water movement, we know that, and to avoid this pressure on a cell mostly store more water inside of the plant cell and applies a turgor pressure on the cell membrane and cell wall to avoid or to minimize or to balance the pressure between environment and the inside of a cell. Okay, so is there any question? Is there any need of very large vacuoles? Yeah, depending upon the function and depending upon the season. Okay, so in a season of a rain or in a rainy season, that time what happen? There is a more water available to a plant. That time more water is placed inside of the vacuoles. At that time what happen? For example, this is a blade of a grass. Okay, this is a blade of a grass. We always seems like there is a droplets, water droplets on this blade of a glass. Okay, so these water droplets we always said that this is because of condensation of a vapor inside of the air. But sometime what happen, the cells which is the part of this grass or blade of grass stores more amount of water inside of the vacuoles, and that time it just release these vacuoles at the edge of the blade, and that's why this water which is extra inside of a cell came out from the cell and hang over the edge of a blade. So it is also the product of your condensation process, but you can't say it is not the product of your plant. It is also a product of your plant. That time vacuoles is larger in a size, and to reduce the water pressure or to reduce the size of vacuole, cell just secretes extra water at the time early morning. Okay, that's why there is a dew like or the bubbles on the uh, grass blade which shines in the first ray of sun. Okay, it looks like beautiful, and we get happy गटेशन. व्हेरी व्हेरी गुड आदित्य. सगळे वर्ड सगळी टर्मिनॉलॉजी तुमच्या पाठ आहे, मला आनंद आहे. ठीक आहे, व्हेरी गुड, इट्स अ गटेशन. ओके, let's see. There are few structures, we call it a microbodies. It is also present inside of the cell. Microbodies are a small sac, okay, bounded by a single layer membrane. So they are bounded by the single layer of lipid membrane. Okay, so they are found in both in plant and animal also. They are classified on the basis of presence of set of enzymes. Okay, so microbodies have a different name depending upon the set of enzyme which is present inside of the microbodies. The first is spirosomes, peroxisomes, glycosomes, and glyoxysomes. So these four vesicles or the sac are comes under the microbodies. Okay, let's see. In a spirosome, we also call it oleosomes. Okay. So it is arises from endoplasmic reticulum layered by a single layer boundary and involved in a storage of fats. That's why we call it oleosome. It is involved in a synthesis of and a storage of fat. That's why we call it oleosome. It is present in endosome of oil seed. Okay, so please take a screenshot of this slide and then write it into your notes. That is what we are going to discuss, Prajakta. Okay, so the function of microbodies. Let's see. A spirosome is a type of microbody. The function of spirosome is synthesizing and storing lipid. Okay, so storage and synthesis of lipid, both job is done by your single microbody, that is a spirosome. That's why we called it oleosome. It is always present in the endosome of oil seed, or you can simply keep in mind that inside of the oil seed. Okay, peroxisome. Okay, why it is called as peroxisome? Let's see. Peroxisome is a busy in a generating and a scavenging activity of hydrogen peroxide is a reason for this name. Okay, so it is a busy to scavenging the hydrogen peroxide. Scavenging means making it less toxic, less toxic. Okay, less toxic, or making it uh, less damageable to our body. Okay, otherwise it will damage our body more. Okay, so due to this hydrogen peroxide, this microbody, we call it a peroxisome, it plays a role in a lipid metabolism also and conversion of reactive oxygen species. Okay, so it is involved in the conversion of reactive oxygen species into the less toxic material. Okay, otherwise it will destroy our cell. So the enzyme which is responsible for the conversion of this S2O2 into H2O. Okay, so S2O2 is converted into S2O. This reaction is not balanced. You can add 2 in both sides, then you will have idea about this balance of reaction. So the enzyme which involve in the conversion of hydrogen peroxide into the water, we called it catalase. Sorry, catalase. So catalase is enzyme which is playing a role in peroxisome for or to neutralize the hydrogen peroxide into the water. Okay. Then glyoxysome. Let's see what is glyoxysome. It is involved in the conversion of fatty acid to sugar. Okay. So conversion of fatty acid to sugar. That's why we called it glyoxysome. Okay. So mostly in a germinating cell, it is present in mostly a germinating cell, and oxidation of fatty acid to acetyl CoA. Okay, so this acetyl CoA is part of your link reaction, which is the a bridge between your glycolysis and Krebs cycle. Okay, आठवतंय का रे काही Krebs cycle आणि glycolysis? Okay, there is a link reaction between glycolysis and Krebs cycle, and this acetyl CoA plays an important role in this reaction as a transporter from a glycolysis to the Krebs cycle. Okay, so because of the synthesis of sugar, we call it a glyoxysome. Okay, then mitochondria. So guys, now we are running a 40-minute late, so we are going to stop here. We will manage one more section for one more session for this the parts of cells. Okay, so what is remaining? Mitochondria and chloroplast is also a part of our cell. We do not discuss, and there is a cytoplasm and cytoskeleton. These two important things also pending by us. Okay, so we will see it later, and these are the part which is remaining from for the discussion. Okay, so we will see it later, guys. For why today we are going to stop here. So guys, if you don't have a subscription, please subscribe our channel with this code in a capital Abhijit. Okay, so this okay, so single subscription will give you unlimited access in our Unacademy app. Okay, so there is a doubt session and the remaining things, which is meeting with your favorite educator is also there. Doubt solving session is also there. This type of facilities are available with a subscription. So please subscribe. If you don't subscribe, so subscribe it first. Okay, so thank you very much, guys. तुमचे क्वेश्चन वाचून मला आनंद होतो. असेच प्रश्न विचारत जा, आपण डिस्कस करत जाऊ. ओके, सो डिस्कशन ऍड्स happiness in our lecture. Okay, so please ask the questions whenever you got any difficulty in the our session. Okay, so